And so, one of the major pathways is to upregulate a very large control system, and that is the upregulation of pKa. Okay, so activation. All right, acronym overload. What is pKa? Protein kinase A, version A. What does protein kinase A really mean? That means it's going to seek out certain proteins in the cell, usually enzymes, but not always, and it's going to phosphorylate them. And we know that there's an association to them because there's a sequence that protein kinase A seeks out. And so we can actually look at the primary sequence of a protein and determine, is it likely to be regulated by pKa or not? All right. Membrane. Signal is on the outside. A signal molecule. It binds to a receptor. Okay. So the signal is going to bind to the receptor. Now this receptor is a G protein receptor. And the G protein receptor has associated to it a G protein. How many parts is in a G protein? Do you remember?
5.
Not 5. Not 4. 3. Good. All right. There are 3 parts to the G protein. Labeled what? Alpha, beta, and gamma. Okay. Now, there is one that is considered R, the regulatory, one that is considered the catalytic or active cellular unit. What are regulatory and what are? Gamma and beta are regulatory. So that's regulatory. The alpha is, okay. In the off, alpha has what bound to it? G what? DP. Okay. But when the signal binds to the receptor, it causes the G protein to dissociate, and the alpha subunit has now a GTP. And that floats over to what enzyme? To stimulate it. Okay. Adenylate cyclase. What is the substrate of the enzyme adenylate cyclase? What is its product? ATP is the substrate. The product is cyclic AMP. Cyclic AMP then upregulates pKa for the catalytic subunits to be released and active. What does pKa? What does cyclic AMP have to do? Bind to R. How many times? 4. 2 in each regulatory subunit to allow the catalytic subunits to go on and cause cellular response. So once the catalytic subunits are released, they are ready to do cellular response. Woo. And we gave you a cellular response example last time as the last thing. What did epinephrine do to muscle cells through this pathway? It released lots and lots of what? Okay, epinephrine, fight, flight, send a signal to the cells. I'm going to release what inside the cells for energy? Glucose from glycogen using the glycogen phosphorylase, which breaks down glycogen into glucose. But there was one regulatory molecule enzyme that we upregulated called glycogen phosphorylase kinase, which turned on and goes through. All right, I said the signal or the domino effect, I want you to know all the way up to getting PKA activated. That is the point of if you have to learn how to drive, I want you to get from the signal to this point. Okay, I'm not going to make you memorize going all the way from New York to Los Angeles yet. I'm going to get you to New York to West Virginia. Okay? That's as far as you're responsible at this moment in time. All right? But don't worry. I'll make you responsible for other things later. All right. That is the upregulation. Let's stop this. Yes?
When the alpha subunit takes on GDP or GTP over GDP, does it drop the beta gamma subunit?
Yes, it releases the beta gamma subunit, which allows conformational change so that the GDP is released and a GTP, the diphosphate, is released, the triphosphate is picked up. All right, this is turn on. Tell me one thing I need to do to turn this totally off. One thing. Okay. Right here, this alpha subunit has a GTP ACE activity, meaning that over time, if GTP is inside of it, it's going to break it down. Once the GTP, the triphosphate, is converted back to the diphosphate, okay, which takes GTP and makes GDP, and so the alpha subunit binds back to the beta gamma, and it's turned off. Okay, that also causes the adenylate cyclase to stop working. Okay, so this did I lose everything? Please no. Okay, all right. This stops the GTPase. Also, that causes this to stop. Because adenylate cyclase needs the alpha subunit of the G protein with a GTP to make it go. I've turned off one part. I make it go backwards here. Okay, that's one of the turn off. There are others. Alpha binds back to the beta gamma. Back to the regulatory. Okay. So I now have no more stimulating G protein, and adenylate cyclase is not making any more cyclic AMP. How else might I stop this?
Block the receptor.
Block the receptor. Okay. What happens is the receptor, once it's stimulated, okay, it causes a conformational change in the regulatory subunit to make this go out. But also, what happens is it will open up a place for it to be phosphorylated. So when it's stimulated, the receptor becomes phosphorylated. Okay. That's one. Second, a protein called beta-arrestin will come over and bind to it. Okay? Once beta-arrestin is bound, the receptor cannot stimulate any more G proteins. So, once the arrestin is bound to the receptor, it's off, even in the presence of signal. Okay, that's two. I can see other places that I need to turn this thing off. What else? Stop the signal. That is not a 3. I have no idea what that is. Stop. Stop it. Okay. Anything else? What's causing cellular response? Isn't it pKa being active? Is pKa still active? Why is pKa still active? Because it's not bound to what? The regulatory subunit, right? Why does the regulatory subunit not bind to the catalytic subunit? Because what? Because it has CAMP bound. CAMP is a concentration. It's a small molecule. It's sitting in the cell. To stop the activation of the regulatory subunit separating from the catalytic, I have to break this down. No! Okay? That is done by an enzyme called ATP. No, not ATP. PDE, phosphodiesterase. The last thing I have to break down is cyclic AMP. And cyclic AMP is broken down by an enzyme called phosphodiesterase. It breaks the cyclic AMP into 5' AMP, or 5' AMP is just AMP. It isn't cyclic. Not having the cyclic form means that it can't bind to the regulatory subunits. If it can't bind to the regulatory subunits, then the catalytic will now bind to the regulatory subunits and be stopped. And so I have to break down cyclic AMP. That's the fourth step. The fifth step is the regulatory subunits bind back to the catalytic subunits on pKa, and that completes that circuit. A little bit more in terms of the beta-arrestin. The conformational change that occurs when the signal comes in, okay? So in this case, it's epinephrine is bound to the receptor. It is able to take the beta-gamma subunit, regulatory subunits, move them away. The alpha is doing its part. But that also opens up a segment in which an enzyme called the BARK, beta-adrenergic receptor kinase, phosphorylates the receptor, creating a new binding pocket in which beta-arrestin, BAR, will bind, and this combination cannot upregulate any more G proteins. In actuality, this receptor, for it to be so that it can be reset, has to be internalized into a vesicle. It's processed, dephosphorylated, the protein is released, and the receptor is sent back to the membrane. It's a really complex process to get that back in. But that is how the receptors are inactivated, even in the presence of a signaling molecule. Okay? So the last pathway, we were upregulating the enzyme pKa. Okay? This one is called the phosphoinositide cascade. Okay? It will upregulate PKC. be protein kinase C. We also saw cyclic GMP will upregulate protein kinase G, and we saw insulin will upregulate protein kinase B. So we've got A, B, C, G, and if you think we had B and we had G, what else do you think? A, B, C, we've got C, so there could be a D, E, F. Okay, there's lots of protein kinases. I believe the A, B, C, and G are the main types of ones that are listed that way. Other ones will have longer names. Okay, let's take a look and see what this cascade is, or this signaling process, and then we'll go through it stepwise.
Binding of a ligand, such as a hormone, to a specific receptor in the cell's plasma membrane activates a trimeric G protein, which in turn activates phospholipase C, which cleaves phosphatidylinositol 4,5-bisphosphate, or PIP2. DAG can diffuse laterally in the plasma membrane, where it encounters protein kinase C, which has moved to the membrane in response to calcium released by a DAG sister molecule, IP3. DAG activates PKC, which phosphorylates many other proteins, leading to cellular responses. DAG is an important second messenger in controlling cell division.
Okay, got it? PLC reacts with PI2 to create IP3 and DAG that goes to the ER to release CA2 plus to upregulate PKC. Oh, Lord have mercy. God help us. All right. Let's see what we know and apply it to this system. A hormone binds a receptor, upregulates a G protein. Wait a minute. That looks exactly like we did before, right? Okay. Hormone receptor G protein. What does the G protein look like? How many subunits? 3. The alpha subunit and the beta gamma are regulatory. Once the beta gamma release and the alpha subunit releases the diphosphate, it picks up a what? A triphosphate. And then it goes and stimulates. In this case, it's going to go to a new enzyme. Before we upregulated adenylate cyclase. I'm not going to do that this time. We are going to upregulate an enzyme called phospholipase C. A phospholipase is lipase, lipidase. We're going to break down a lipid. We're going to break down a lipid that has a phosphate on it, phospholipase. Okay, and it's C is where it's going to break. Okay, we're going to break a membrane-bound protein. A membrane-bound lipid. The lipid it will break down will break it and create two things. Here is the lipid. It has 2 acyl groups, long fats. It has the sugar inositol on it. It has 2 phosphates on the sugar itself, and the lipid is a phospholipid, so that phosphate is part of the lipid. Okay, so inositol, phosphoinositol, okay, and then diphosphate is there. That gets broken down, and phospholipases, there's different versions of it. All right, phospholipase A breaks it down there. B or A2 breaks it down here. Let's see, C will break it here, but this is the one we want. Oh no, C breaks it down there. D breaks it down there. We want C, and so it's going to break it into 2 things: diacylglycerol. Glycerol, that molecule, and 2 acyls, diacylglycerol, and an inositol that has 1, 2, 3 phosphates, which will make it IP3, inositol triphosphate. Okay, so I've broken down a membrane, 2 pieces. One is still floating in the membrane. because it can't float away, but the IP3 now becomes soluble and can diffuse inside the cell. Okay, so the enzyme is upregulated PLC to make DAG and PI3. Woo. All right, DAG and IP3. The inositol 3-phosphate floats to a receptor in the ER. Okay, so this is the ER, and ER is full of calcium. Once it hits the receptor, opens up a channel because of a high concentration on the inside, low concentration on the outside, calcium floods the cell. Calcium is considered a second messenger. IP3 is a second messenger. DAG is created as a second messenger. Now, I have DAG and calcium floating inside the cell. It will upregulate the enzyme protein kinase C. And what happens is the DAG will bind to the protein kinase C, the diacylglycerol, and increase its affinity for calcium. Calcium then effectively binds the protein kinase C very highly, and the protein kinase C becomes active, leading to cellular response. Protein kinase C will phosphorylate lots of enzymes, lots of things inside the cell. Will protein kinase C phosphorylate everything that protein kinase A does? Why does A and C phosphorylate different things? Because different consensus sequence. Each protein has a place where the kinase will bind to. Are there proteins that are activated by both protein kinase C and protein kinase A? Yes, they're phosphorylated at different places, but most enzymes are either or. Okay. Turn it off. What do we have to do to turn this off? Well, we got to get rid of everything that was created. Okay. Was PKC created in this pathway? No, but it was stimulated. What do you think stimulated it? IP3 and DAG, but IP3 not directly, IP3 through calcium. So to stop it, we got to get rid of DAG. Okay? What we do is we break it down, or we add a polar head group. As long as we convert it, it stops stimulating. So this is one. Calcium has been released. So if it's released, what do we have to do? We have to block it, or we have to, where was it first? We've got to pump it back in. That would require a pump that uses ATP to pump against the concentration difference. But the problem is, if we pump right this very moment back in, what's going to happen immediately? It's going to come right back out. So what do we have to do to close the receptor? We have to break IP3. We either dephosphorylate it to have 2 phosphates, or we phosphorylate it up to have 4. As long as it goes either way, it's not going to bother us. So if we change this to IP2 or phosphorylate it to IP4, the channel closes. And so the pumping, which is step 2, now becomes effective because 3, I've gotten rid of IP3. But the problem is IP3 only created once? IP3 and DAG are made by what? By the phospholipase C. How was that stimulated? What made the phospholipase excited? It was touched by the G protein. Okay, and so now we've got to stop that. Okay, and the alpha subunit of G proteins have what activity? A GTPase activity. So it turns itself off. Goes back and binds to the regulatory subunits. Stops PLC from being. What happens to receptors? What did we just say with the PKA? As soon as it stimulates G proteins, what does it open up on its other part? A sequence that can be phosphorylated, and a protein called what? Arrestin binds and stops the receptor from being able to be stimulated. And last but not least, you have to stop the hormone from being produced. That will stop this whole process. Okay? And what's good about this is I know I'm recording. Okay? So the protein kinase C in the inactive form, the calcium concentrations are very low, and there is a pseudo substrate in the active site. PKC has its own little piece of protein that fits into the active site and blocks anything from being bound. DAG binding to PKC makes it more susceptible to calcium. Okay? And when calcium binds, that pseudo substrate pops out. What we really do, all of that regulatory event is just to make sure that I open up the active site so things can get accessed. And then it'll phosphorylate lots and lots of targets. It is a serine-threonine kinase. What targets will it activate? Well, that will depend upon what tissue type it is. In the DNA, all proteins are available, but in each tissue type, only certain proteins are produced at high levels. And so I might upregulate PKC in one cell type and phosphorylate a whole host of proteins, but in another cell type, I'll phosphorylate other proteins just because in different cell types, the concentrations of what's available are different, which cause different functions. IP3 has to be dephosphorylated or phosphorylated into something different. DAG is phosphorylated or broken down. And once that's gone, I can't stimulate. Calcium is sequestered back in the ER through pumps. The G protein has its GTPase activity. Once it's broken down to the diphosphate, it binds to the regulatory subunits. And last but not least, the beta-arrestin on the receptor itself. So I probably should put that in. The arrestin binds to the receptor after phosphorylation. Okay, and then the last thing, the hormone must, don't know why I put a W, must stop. All right, turned it all off. Cyclic AMP, a very powerful second messenger. Okay, but we saw calcium as a second messenger. Calcium is a huge messenger within the cytosol. Most of the time, calcium is going to either bind directly to something to upregulate it or bind through a sensing protein. Most cellular events inside the actual cytosol, it's very, very low in calcium. We control this very abruptly, a very controlled measure about what calcium concentrations are inside the cell. It's about 100 nanomolar, normally accomplished by the the calcium ATPase and the sodium-calcium exchanger. We looked at those pumps before. The steeper gradient allows very quick changes in the calcium concentration, which is a rapid type of regulation. Most of the time, calcium is signaled through a binding protein. Okay? And usually, in the presence of calcium, proteins are functional. In the absence of calcium, most of the time the protein is inactivated. The protein that senses calcium is called calmodulin. It's a very small protein. It binds 4 calciums per molecule of the calmodulin, or CAM. The calcium is bound both in calmodulin and in many types of proteins in what's called now a helic loop, it used to be called a helic loop helic motif. Now the helix loop helix motif is a DNA binding motif. So we now call this the EF hand. The EF hand is because the E helix looks like, if you make your finger, this would be the E helix, this would be the F helix. And so the E is the finger, the F is the thumb, and the loop is where the calcium sits inside. And you get really tight binding, very, very good binding constants. The calmodulin, once bound to calcium, has a large conformational change. And what it behaves like is this. All right, it's floating around kind of a pocket up here, a pocket down here. When calcium binds, it actually turns and it clamps around the protein, an alpha helix of a protein. And when it's clamped around the protein, it will upregulate it. Once calcium is released, calmodulin opens up and it falls away and it isn't bound to the protein. So lots of proteins are regulated by calcium binding to calmodulin and then calmodulin binding to that protein, making a complex that is upregulated. Okay. All right. One more signaling process. Okay. Up to this point in time, we've talked about a hormone basically released in the bloodstream. It goes to a cell, it binds to a receptor, and causes an event process to happen. All right. Very similarly, your vision works just like one of these pathways. Okay. And so, but the signal is a photon of light. Which is kind of oddball. How in the world is a photon of light actually going to cause a change in the cellular response? Okay, and so we'll see about that. Vertebrates have two kinds of photoreceptors. We have cones and we have rods. Cones will detect color and bright light. Rods do not perceive color and are ones that are optimized for dim light. All right, debate. Do dogs see color? Vertebrates have the ability to have those types. Not all vertebrates have them. Okay, how in the world? Some people say dogs don't. What, do you speak to a dog psychologist to find out what they feel about color? Or do they have very little cones or no cones and only rods? And so the debate is yours. Okay, we're going to look at the rods, and we're going to see that rods can actually be activated by a single photon of light. What it will do is cause an electrical signal, which is what? Very simply, across a membrane, we're going to have what flow? Ions. Okay. The signal will process through the retinal nerve to the optic nerve to send a signal to the brain. All right. Now, we know exactly how the signal is generated. We absolutely have no idea how our brain actually processes that to make us perceive the information that we see. All right. Does this look like a typical cell that you see at the beginning of a biology textbook? No. But this is a cell. Okay. Do you see anything that is familiar to you? Mitochondria. Yes, it has mitochondria. Because that's the powerhouse of the cell. Okay. Yeah, there's the ER there. A nucleus. That is great. All right. What about this end over here? Have you seen something similar to this end? That's like an axon. And right here, vesicles that hold acetylcholine be very similar. Okay. What about up here? Does this look like anything that you've seen before? Okay. What was that? The heat things? Oh, yeah, that's right. Elements. Hot dog rollers. Okay, I opened myself up here. Okay, those are called. They're discs located in the rod, and you can see why this cell is named the rod. It looks like a rod. Okay, what this does is a disc, kind of like, if you look down, it'd be round. If you look on the side, it looks flat. Kind of looks like the shape of a red blood cell. Round, kind of dipped in the middle, a disc. What this does is it gives us a lot of surface area of membrane. The detection of the light signal will be in a protein that is embedded in a membrane. Okay? So we have lots and lots of membrane for the chromophore, the light-absorbing group, okay, which is called rhodopsin. It is a protein. Okay, so that's the chromophore. Rhodopsin is a protein that is the protein opsin plus the organic material called retinal. In particular, the 11-cis retinal. 11-cis retinal is a byproduct of vitamin A. Okay, vitamin A is an organic molecule that we have to eat. All right, vitamin A can be derived from something that we eat. You know what? Something that we can eat that can make vitamin A?
Carrots.
Carrots, yes. Carrots are that. What is in carrots? I want to be a little bit more specific than carrot. What is it? Beta carotene. Beta carotene is a very long structure in which, if you actually split it in the middle, you get 2 vitamin A components. Okay. Why are these molecules good for vision? What are we trying to capture? Light. And what was that? Conjugation. One double bond will absorb energy. But what range of energy will it absorb in? The UV range. All right? That's not where we see. But if you add conjugated double bonds, you move the maximum absorbance from the UV into the visible range. Okay? And so having the conjugated double bonds allows things to absorb light in the visible range, also to have color. Okay. Now, for us, there's this thing about carrots. What is the wives' tale about carrots? It helps you see better. So, if you have blurry vision and glasses, you eat lots and lots and lots of carrots, yes? That will help your vision. No, it doesn't. It may make you look orange, but it doesn't actually improve your vision in terms of sharpness. Why in the world do lots of us need to wear glasses or contacts? Because our eyeballs are misshapen, and the light comes in and it's not focused right. Okay? And so we have to have corrective lenses so it helps our eyes when it refracts and reflects light. We have to do it beforehand so we will get a sharp image. Has nothing to do with eyesight that way, but it has everything to do with eyesight in terms of something called like night blindness. Okay, have you ever driven down the road and someone with their high beams comes up and unfortunately you look at the lights instead of looking at the line, and all of a sudden what can't you do? You can't see. What you do is you overwhelm the number of sensing molecules that you have, and you don't have enough. You don't have enough because you didn't eat enough of the vitamin, you didn't eat enough carrots. So the more you have of the vitamin, the more opportunities or the more molecules can absorb the photons of light. And I'll talk about why that night blindness actually occurs in just a bit. Okay. Now, the vitamin and the retinal, this is the 11-cis retinal. Is there differences? Do they look the same? No. What is different? transist. Essentially, that is the biggest piece of the puzzle for why vision can occur. Also, the alcohol is converted to an aldehyde. The aldehyde allows us to actually link it to the protein. Okay. The protein itself has a optimal lysine, and the nitrogen of that lysine and the aldehyde of the retinal will react to form what's called Schiff's base. I actually have to covalently link that organic molecule to the protein. And so I link it to one amino acid, and just like in hemoglobin, if I move that one amino acid, I change the entire shape of the protein. Okay? And so that organic molecule is covalently linked to the protein, ready for excitation. All right. So, what is going to happen? This is the magic of light. Light comes in, and it causes a transition from the cis to the trans upon absorption. And the cellular response happens. Now, think about this. The protein is connected here, right? Because I'm at the end, and it's bent in the cis. Once the energy of the photon is absorbed, it causes that change from cis to trans. That change is relatively a very large movement, even larger than the movement of that iron when oxygen binds. This is a huge change, which makes the lysine move a lot, which then moves this protein a lot. It goes from a unexcited or resting state to the excited form. The protein changes shape. The rhodopsin changes shape. Upwards of 5 angstrom movement. Okay, here's the deal though. This protein now is excited, but it cannot be excited again, ever, in this form. I actually have to break the protein down by removing that organic molecule, manipulating the organic molecule through reactions, and putting back a cis molecule in. That takes time. After the isomerization, lots of things occur. Okay, there are multiple intermediates. Okay, that are observed in that full transition. For us kind of to reset, the retinal has to be broken apart, diffused from the opsin. The retinal has to be reduced to retinol. The alcohol can then be converted in the dark back into the 11 cis by oxidation to from the alcohol back to the aldehyde, and then it can be reassessed into the photoreceptor in the actual protein, and then it's ready. All right, here's the deal. If you look at the bright lights of an oncoming car for just a moment, you become overwhelmed, and you have to then wait for all of those organic molecules to be broken off, reduced, isomerized, oxidized, and be put back in. And it takes time. If you don't have a lot of them in the first place, then you can overwhelm it, and it takes a long time for that to come back. If you're driving at the speed limit, like anybody ever does, or above the speed limit, for a while you're not seeing and you're driving. This is not a good thing. Okay? And so, if you have more of the organic molecule by eating a good, healthy diet of vegetables, or if you look on the side of the cereal box, all of this vitamin A, then you will have enough to be able to see. Now, one time, between my freshman and sophomore year at college, my parents were living in San Diego, flew back, and I had to get a job. And so I decided, okay, I went down to SeaWorld to get a job. And I got hired at SeaWorld. I got hired at SeaWorld to do spotlights, pyrotechnics, and lasers. And so I spent a summer doing all of that. Now, the laser show is pretty cool, although when you're adjusting the light box where you have to turn on the laser a little bit, don't let your buddy turn it up even more powerful because it will burn a hole through your shirt. Learn that. Spotlights are pretty cool, although the one show song that you hear 4 times a day for all summer, I will never like those songs again. Pyrotechnics, I was never on that team. Running around in those floats, driving a car with all the people in the back is really hard. But there was one job in which was called show flow, where I sat in a room, and when a show stadium got full, I would call over and we would then start directing people to other. It's the most boring job in the world, except for the fact that, have you ever seen those big lights that go in the night sky? My job was to turn that on. Okay. One day when I was being trained, I was told not to look into those. Okay. One day, 2 days, 3 days. Then one day I turned it on and happened to glance down at the light. And I will tell you, every single rhodopsin in my eyeballs were stimulated. To the point that I was actually blind for a good couple minutes before it actually come back, my eyesight. So you can overwhelm, even if you have a lot of rhodopsins, by being overstimulated. Okay? And it just took time for those to be reset. Okay? And so we have now started that. So PKA, know how to upregulate that. Know how to upregulate PKC. Know this. We're going to do vision. Okay? And insulin. Turn on and turn off. But what are you seeing amongst these? Even though you need to know this part seems to be the same for all of them, or they're very similar steps. So you can make generalizations about what you see. Okay? And just think, we're going to do vision. Well, what other senses you have? We're going to learn how you smell. Okay, how smells happen. Okay, taste, we're going to look at those too. And so we're going to look at some of these, but again, they're going to be very similar. There's some similarities in what's going to happen. It's a weekend. Why are you here?